The food we eat are actually parts of food chains. Plants are eaten by worms, worms are eaten by chicken, and chickens are eaten by humans. Therefore, the energy that humans receive from our food ultimately comes from the plants. Plants make their own food using photosynthesis. Through photosynthesis, plants use the energy from sunlight to transform carbon dioxide and water from the environment into glucose and oxygen. That glucose are then transformed into more complex organic molecules to form leaves, stems, roots, fruits, and others. These plant components form the basis of the food chain, like this example. Photosynthesis consists of two stages, light-dependent reaction and light-independent reaction. The second stage is called light-independent reaction because the light energy is not directly required in this reaction. The light-independent reaction, also known as Kevin cycle or C3 pathways. Kevin cycle is crucial to convert CO2 into complex organic compounds mentioned before. Nevertheless, the two key products from the light-dependent reactions are needed to make the Kevin cycle works, ATP and NADPH plus H+. You may recall that both ATP and NADPH plus H+ were made during the light-dependent reactions at the thallic membrane. The Kevin cycle takes place in the stroma of chloroplasts of C3 plants during daylight. ATP is the source of energy. Meanwhile, NADPH plus H+, as reducing agent, provides the electrons and hydrogens that are needed to reduce CO2 into organic molecules such as sugars in the Kevin cycle. There are three phases in Kevin cycle. Here's how it works. Phase 1, carbon fixation. An enzyme called RUBP carboxylase oxygenase, or known as Rubisco, catalyzes the uptake of CO2 from the air and combine it to a 5-carbon compound called ribulose biphosphate, RUBP. This makes an unstable 6-carbon intermediate molecules that immediately splits in half to form 2 molecules of 3-phosphoglycerate. Notice that we did carbon fixation for 3 molecules of CO2, so we will have in total 6 molecules of 3-phosphoglycerates. You will see why soon. Phase 2, reduction. Each 3-phosphoglycerate receives 1 phosphate group from 1 ATP, forming 6 molecules of 1,3-bisphosphoglycerate. The phosphate group comes from the hydrolysis of ATP, so in this reaction, 6 ATPs are required. Then the 6 molecules of NADPH plus H+ donates their electron to reduce the 6 molecules of 1,3-bisphosphoglycerate. The 1,3-bisphosphoglycerate also loses the phosphate group, becoming 6 molecules of glyceraldehyde 3-phosphate, or more commonly called as G3P. 6 NADPH plus H+ are then oxidized to 6 NADP+. G3P is the starting point for the synthesis of other organic molecules. Notice that for a net synthesis of one molecule of 3-carbon G3P, it requires 3 times of Kevin cycle and hence 3 molecules of CO2. 3 carbons in, 3 carbons out. Makes sense, right? Phase 3, regeneration of RUBP. Notice in our model that only 1 molecule of G3P exits the cycle, but 5 more remain to continue the cycle. These 5 molecules of G3P are rearranged to regenerate our starting compound, RUBP, through a series of complex enzymatic reactions. Remember, the Kevin cycle is a cycle, so we need to make our starting compound again to keep the cycle going. Since we have 5 G3P molecules and each one has 3 carbons, so in total we have 15 carbon atoms. And since we have 15 carbon atoms and we want to use them to make 5 carbon RUBP, so we can make 3 RUBP molecules. To do this, energy is required in the form of 3 ATP molecules. Finally, 3 RUBPs are regenerated and ready to enter the cycle again. Now that we've completed the 3 times of Kevin cycle to produce 1 molecule of 3-carbon G3P, let's go through the process again so that we can make 6-carbon sugar. Remember, our overall photosynthesis equation has 6 CO2 being used to make 1 molecule of 6-carbon glucose. Thus, we need 2 G3Ps to make 1 glucose. So, in total, 6 CO2 are needed for carbon fixation in 6 times of Kevin cycle. Since fructose is isomer of glucose, glucose and fructose are then joined to form sucrose. Now you've seen how the Kevin cycle is used to make glucose and how dependent reactions provide the NADPH plus H plus and ATP that were needed to make the Kevin cycle works. The NADP plus and ADP formed in this process are then returned to the thylakoid membrane to regenerate NADPH plus H plus and ATP in the light-dependent reactions. The glucose and other organic compounds made as a result of this process in photosynthesis are what allow plants and algae to grow and support practically all life including you.